Namaskar. As we are going to discuss bacterial reproduction, transformation, transduction, and conjugation. In this module, we will discuss about the part 2 transduction mechanism. In BSc semester 1, in the subject biotechnology, in the paper General Microbiology, this topic is being discussed. After studying this lesson, you will be able to define transduction in bacteria, describe the process of transduction in bacteria, explain the importance of transduction, differentiate a specialized and generalized transduction. So these are the objectives of today's session. As we have already studied in part 1 of this e-content, that is bacterial reproduction and the mechanism of transformation, in which bacteria take free DNA from their surroundings and incorporate into their genome. Now, we are going to discuss transfer of bacterial fragment from one bacterium to other, which is mediated by bacteriophage. As we know that bacterial reproduction includes transfer of genetic material between bacterial cells, allowing them to adapt and develop, and allowing them to thrive in the diverse environment. Bacterial transduction, the transfer of a small portion of bacterial genome from a donor to recipient bacterium through bacteriophages is called transduction. Transduction is a mechanism of genetic transfer through bacteriophages. Transduction was discovered in 1950s by Joseph Lederberg and Norton Zinder. Zinder and Lederberg in 1952 found in their research that the P22 type of bacteriophage in Salmonella typhimurium can transfer a portion of bacterial DNA from one bacterium to another. All the phases involved in transduction are temperate, those that either inactivate the cell at once. They can multiply inside the bacterial cell and eventually cause lysis of the host cell or bind with bacterial DNA to remain in the host cell. A bacterium that harbors temperate phases is called lysogenic. It can be general or specific. In generalized transduction, almost any part of the donor's chromosome can be transferred, but in when it is specific, as in the case with lambda prophase in strain K12 of E. coli, only a specific loci, that is gall and biogenes, are transferred. Bacterial transduction occurs in several steps, including infection of bacterial cells by bacteriophages, packaging of bacterial DNA into phage particles, and transfer of this DNA to recipient bacterial cells. This process plays an important role in our understanding of bacterial growth, a spread of antibiotic resistance, and genetics. The second group of prokaryotes other than the bacteria, that is archaea, is not infected with bacteriophages, but it has its own virus that carries genetic material from one person to another. Now, the process of bacterial transduction. You can see here the bacteria 1 having the bacterial DNA. The first step is attachment and infection. During bacteriophage infection, bacteriophages inject their genetic material into the bacterial cell. First of all, bacteriophages inject their genetic material into the bacterial cell. They have their specific receptor on the surface of the bacterial cells. Each bacteriophage contains a receptor binding protein or tail fiber that recognizes the specific receptor. Once attached, the phage injects its genetic material into the bacterial cell, handling the cell's machinery for its replication. In other words, bacteria hijack the cellular machinery. Now, the replication and assembly. Inside the bacterial cell, the phage genome is replicated and new viral particles, that is the phage offsprings, are assembled. This replication of phage DNA is done with the help of bacterial enzymes. This is an important step in the phage life cycle and it ultimately leads to the release of phage particles. During the assembly of new phage particles, mistakes can occur, but this bacteria can replicate along with this phage particle. That is, the bacterial DNA replicates as well as the bacteriophage DNA, which is already incorporated into bacterial DNA, also replicates along with the bacteria. But during the assembly of new phage particles, mistakes can occur. Instead of just packaging the phage genome, phage can mistakenly package a piece of bacterial DNA along with phage DNA into the phage capsid. This error leads to the formation of transducing particles which contain both phage and bacterial DNA. These defective transducing phases particles, also known as transducing phases, can also infect another bacterial cell. Transducing particles, which now contain a piece of bacterial DNA, is transferred from one bacterium to another bacterium during transduction. Now, this new phage particle having the bacterial DNA of bacteria 1 can be transferred to the bacteria 2 DNA. In this way, transduction can occur. Now, this transduction may be of two types. First is generalized transduction. In this type, any bacterial DNA can be transferred to the recipient cell. This occurs when phages mistakenly package random bacterial DNA during viral assembly. This means that transducing particles can carry gene or genetic material from donor bacterial cell. Now, a special transduction. In this type of transduction, a specific genes can be transferred near the site of integration of phage DNA into bacterial genome. It involves the transfer of a specific genes or genetic sequence that are located near the site where the phage bacteria integrates into the chromosome, that is prophage. It occurs when phage bacteria stimulate themselves from chromosomes, bringing together adjacent bacterial genes. Now come to the importance of bacterial reproduction. The first is genetic exchange. That is, bacterial transduction facilitates the exchange of genetic material between bacterial cells. Rapid growth. It has the potential for rapid spread of genetic innovations in the population as well as enabling bacteria to adapt in various situations. Antibiotic resistance prevalence can be developed in bacteria through transduction. Transducing particles can transfer antibiotic resistant genes. Genetic mapping. By tracking the transfer of specific genes through transduction, the locations of these genes on bacterial DNA can be mapped and their roles can be understood. Phage therapy. A specific bacteriophages can be used to target and kill pathogenic bacteria. This approach has led to it being seen as an alternative to antibiotics. If you summarize all of the transduction process, the first one is bacterial transduction is a process by which bacteria transfer genetic material through bacteriophages. This process involving generalized and specialized transduction has applications in bacterial genetics and evolution and the spread of genetic traits including antibiotic resistance. The study of bacterial transduction leads to an understanding of the complex process occurring between bacteria and their viral counterparts. An understanding of these processes enhances our insight into bacterial genetics and evolution, as well as valuable insight into combating antibiotic resistance, new treatment practices, and harnessing their capabilities for human benefits. Students, you can refer these books for further reading of the transduction. These are textbook Pretora and Funke ACL, that is Microbiology and Introduction, 9th edition Pearson Education. Some of the reference books are there by the Alexopoulos and Mims, by J and Listener and Golden, by Matinko and Parker, by Pearson Benjamin Cummings, by Piljar, Chan and Krieg, by Steiner, Ingram and Willis Penter, Willie and Steerwood. Further reading can be done through the video resources on these websites. You can see here, these are the here on these websites, video resources are available. Text resources, if you see, these are the text resources, that is Genetics, Medical Microbiology by NCBI Bookshelf, Transduction and Information by Springer Link, Exchange of Genes, Transformation, Conjugation and Transduction, that is Dynamics of the Bacterial Chromosomes, Bacterial Genetics, Conjugation, Transduction and Transformation, and Bacterial Recombination, that is in PubMed literature. Now come to the assessment. Which of the following is responsible for carrying out bacterial transduction? The answer is bacteriophages. What is the transducing particle in bacterial transduction? The answer is a phage-infected bacterial cell carrying a fragment of bacterial DNA from the donor cells. What is the main difference between generalized transduction and specialized transduction? The answer is the specificity of the DNA fragments transferred. Which type of transduction involves the accidental packaging of bacterial DNA fragments into bacteriophages? The answer is generalized transduction. Bacterial dash is the process where genetic material is transferred from one bacterium to another by a bacteriophage. The answer is transduction. Students, we have discussed today about the transduction. In previous e-content, we have discussed about the transformation. So in bacterial reproduction, we have discussed transformation and transduction. In next e-content, we will be discussing about the conjugation process. Thank you.